cell proliferation without any outside signal. So we call it as cell sufficiency in growth signal. So they have insensitivity to growth inhibitory signals. Means if the tumor suppressor genes send a message for them to stop, if there is a growth inhibitory signal for them to stop, they don't respond. So they have insensitivity to growth inhibitory signals. So again, they continue their proliferation. They can evade from the cell death. Remember, we had apoptosis as a programmed cell death, but the tumor cells, the cancer cells, they escape from this mechanism and they continue their proliferation. So we say that they don't respond to the process of apoptosis. They have limitless replicate potential. I mean, they can divide continuously without any limitless. There is process of angiogenesis. Remember, we said that tumor cells, if they want to continue their life, they need the blood supply, they need the nutrition. So they do angiogenesis by their own. They create new blood vessels. They provide for themselves new blood vessels and blood supply and nutrition. And because of that, they can continue their life because they already have blood, nutrition, so they can continue their proliferation. They have ability to invade. Remember, we said that the tumor cells, those that are malignant, they can destroy, they can invade, and they can go to the lymphatic channels, they can go to the hematological pathway, the blood circulation, they do angio invasion. So when they invade to the blood vessels, they can go to the distant side. For example, we have breast cancer, they go to the lymphatic channel, and after that they go to the lymph node. And later on, maybe they can show angio invasion. They go to the blood circulation, and because of that, they go to the brain. Because we don't have a lymph channels in the brain. We have angio invasion, and we have a patient suffering from breast cancer, and later on coming with a brain cancer. And this brain cancer is a metastatic from the breast. Or, for example, we have a case of a lung cancer, later on coming with a mass in the adrenal. Because it's an angio invasion, there is a risk of metastasizing tumor of the lung coming to the adrenal. So they have a specific pathway. They can metastasize to different distant sites. So this is also another hallmark of the cancer. They can reprogram the process of the energy metabolism. They can provide for themselves energy, blood supply, nutrition, so they can reprogram. They can evade, they can escape from our immune system, because at the normal condition, our immune system, they recognize them as outside antigen, try to actually confine them and kill them, but since they are very, very smart, they can escape from our immune system. They change the antigen at the surface. They maybe show a subtle antigenicity, and because of that, our immune system cannot recognize the tumor cells, and they can escape from our immune system. They have a genomic instability. Maybe today, coming with the breast cancer, we treat the patient, and patient shows complete response, but later on, after 6 months, 10 years, as she just mentioned, patients come in with a recurrence or come in with a relapse. Because the tumor shows genomic instability, they undergo further mutation, further additional changes in the genomes, and they have, because they have genomic instability, they can change the process of the genes and come in again with different profile of the genes. So because of that, we call it that's a relapse. Even though we treat the patient, even though the patient shows complete response, but again coming with the relapse of the cancer because of this process of genomic instability. And then the tumor promoting inflammation. When they promote the process of inflammation, again they have a risk to escape from our immune system. This is the picture, shows that the tumor cells, they have this hallmark. As we mentioned, cell sufficiency in growth signals. They have insensitivity to anti-growth signals. They have invasion, metastasizing, and they have limitless replicative potential, sustained angiogenesis, and they can evade from apoptosis. So, carcinogenesis is a multi-step process resulting from the accumulation of multiple genetic alterations that collectively give rise to transform phenotype. What I mean, maybe initially you started with only one mutation. This one mutation, we call it, for example, clonal hematopoiesis. But this mutation is not enough to get the cancer. Maybe if they do genomic sequencing for those people that they are sitting in the class, many of us, they have some subtle mutation. But this subtle mutation, even though it's first step, is not enough for us to getting the cancer. But if exposed to the different exposure, for example, sunlight or different chemical drug, and later on additional mutation coming, additional deletion coming, additional maybe chromosome changes coming, this progression cause we have a more chance to getting the cancer. Initially, maybe all of us have some clonal hematopoiesis abnormality or instability in some genes, but this is only maybe small. It's not enough for that to show the cancer or tumor. But later on, when this progress and there is additional and genetic changes, we have a greater malignant potential of this mutation. So increasing malignancy often is acquired in an incremental fashion. At the molecular level, tumor progression and associated heterogeneity are more likely to result from multiple mutations that accumulate independently in different cells and generating subcolon with different characteristics. During progression, tumor cells are subjected to immune and non-immune selection, and after that, the tumor cells select those genes that they are more strong, they can continue to their proliferation, they are not going to recognize our immune system, and because of that positively selected genes, they can continue to the cell proliferation. For example, we have a normal cell here, normal cell, and we have a carcinogen-induced changes in normal cell change, become tumor cells. See that there is monoclonality, they are all same as a red color. So this is, since there is monoclonality and proliferation, there is risk of the cancer. But these tumor cells undergo further mutation, and see the color is changed because we have a proliferation of genetically unstable cells. They have more genetic mutation, addition, deletion, and see that there is different color as a purple, pinkish, and there is clonal expansion of surviving cells that they are vary in terms of mutation and additional genetic changes. See that those that they are more strong, they can continue their life, they can escape from our immune system, they have this potential to grow, and later on we have a clonal expansion of these surviving cells, and they have different potential. Some of them they can metastasize, some of them they can invade, they can escape from apoptosis and immune system. So this is the process of tumor progression. So for example, I mentioned we treat the patient, but the tumor relapse means occur after chemotherapy. Initially, patients show complete response to the chemotherapy, and patients become okay. But later on, again, the tumor is coming, and we have a process of relapse. The recurrence of the tumor is almost always resistance to the drug regimen that, before this we gave to the patient. Maybe initially you treat the patient with this simple drug, and patient shows complete response. If the tumor relapse, if you give the same drug, they are not going to respond to this drug because become resistance to that drug. This acquired resistance is a manifestation of the selection. Those subcolon of the genes that they are in the tumor or in the cancer, that they have a chance to escape from our immune system, escape from the apoptosis, escape from the process of apoptosis, they can survive and responsible for the tumor regrowth. This is a genomic evolution and genomic selection, and those selected genes, those involuted genes, they have tendency to become more aggressive they have tendency to become less responsive to the per-use therapy. So we need to have a specific new drug for this patient, or another generation of the drug for these patients to treat them. Okay, for hallmark of the cancer, self-sufficiency in growth signal. We have learned already the process of the growth factor receptor, and that. This explains those processes. Under physiologic conditions, cell proliferation, we mentioned, is under the process of growth factor receptor, growth factor. Remember this picture we have learned. Now something wrong happened, and transmission of the transducted signal across the cytosol to the nucleus is not under the control. And it's not anymore under the control of the physiologic condition that we have in the cell cycle. So cells become actually because automatically they can divide without any signal from the outside. They are not tightly under the control, and they proliferate. We call it they have self-sufficiency in growth signal. In the normal condition, all normal cells require stimulation by growth factor to undergo proliferation. Most soluble growth factor are made by one cell types, we call it, by neighboring cells, we call it paracrine action. But many cancer cells acquire growth cell sufficiency. They don't need actually signal from the outside to do the process of the cell proliferation. They do by their own. They activate their own. They don't need outside signal to do the process of proliferation. So we call it cell sufficiency in growth signal. We mentioned PDGF, TGF alpha in glioblastoma is a malignant brain tumor sarcoma. Cancer cells acquire growth cell sufficiency by interaction by stroma. In some cases, the tumor cells send signal to activate normal cell in the supportive stroma, which is produce growth factor that promote tumor growth factor. So how does the tumor cells become actually self-sufficient in growth signal? Could be as a mutation or overexpression of genes that they do the process of the proto-oncogene. Remember we mentioned breast cancer as a HER2, EGFR, or even lung cancer. Could be a downstream signal production. We mentioned the RAS, ABL. RAS is the most commonly mutated proto-oncogene in human cancer. Approximately 30% of all human tumor cancers contain the RAS genes. RAS, we mentioned on the table, remember RAS commonly is mutated in human cancers and is important in the groups of oncogenes. We have a normal cell cycle that you are already familiar with that, and this cell cycle is tightly under the control. We have a signal that as a checkpoint for each step, like as those that they want to process from the G1 to the to S to G2 is under the control. When something wrong happens in the cycling, for example, in the cycling dependent kinase, the cells is not under the control, the cell proliferation is not under the control of the cell cycling, and as a result we have a cell proliferation and tumors. This is for the cell cycle. Okay. The second group we say that's insensitivity to growth inhibitory signal. This is the process of the Knudsen, that you show that we have a Knudsen phenomenon. Knudsen phenomenon in proposed in new famous two hits hypothesis. What I mean by that, remember the RB gene. RB gene is, we mentioned, is a tumor suppressor gene. We mentioned it's a governor gene, as it's responsible for retinoblastoma tumor in the childhood. And we mentioned this RB gene is responsible for RB protein, and the gene is located on chromosome 13. What is the Knudsen phenomenon? Knudsen phenomenon or two-hit hypothesis mentioned that we need two mutations or two hits to produce retinoblastoma. I'll show you in the picture. So we have two pictures here. One is, see here, sporadic forms. Okay. So you see that here we have two somatic cells of parents. This is for father and mother, and germ cells are affected. This is a zygote, and here see that at the normal condition somatic cells of the child, and maybe if let's say if only one parent carry these genes, I'll show it to you here. So we have two forms, we have familial and sporadic. In familial cases, children inherit one defective copy of the RB gene as a germline. When we say the germline, means all the cells they have these affected genes. So retinoblastoma develop when the normal RB gene is lost in retinoblast as a result of the somatic mutation. They have one autosomal dominant inherited pattern. Only one single mutation is required for expression of the disease. In sporadic cases, both normal are allele affected during the life, or they lost during the life. A cell heterozygosity and the RB locus is not an neoplastic. Tumor develop when the cell loses its normal RB gene copy and thus becomes homozygous for the mutant allele. I show you how we call it loss of heterozygosity. See here. Sporadic. So this is a familial. So the familial, see that we have a mother and father. For this one, we have a one mutant RB genes. So when the child born already has one affected genes, this pinkish one, and another one is okay. It's okay? So the second mutation happened during the life. So we have one from parents, another one happened during the life. We have one inherited, another one acquired during the life. When both affected, now we have retinoblastoma. So we call it loss of heterozygosity, because now there is heterozygote. One is affected, another one is a normal. But when there is loss of heterozygosity, means both become affected. So we call it Knudsen phenomenon because it's a loss of heterozygosity. Here, still, even though there is a chance to get the cancer, it's not enough. We need to have one more affected genes to get the cancer, become mutated. So this is a loss of heterozygosity. This is a familial form. But for the somatic one, they are normal when they born. Both genes are okay. Both genes are okay, and there is no any mutation here. Now, one happened during the life, become pinkish. Another one again happened, so we have two somatic mutation during the life. This is also loss of heterozygosity, and again we have a retinoblastoma. So could be as a familial or could be as a sporadic, but we need both allele affected to get the cancer. This is we call it Knudsen phenomenon or loss of heterozygosity. Means here still there is heterozygosity. When the loss of heterozygosity happens, we have the cancer. So this is a Knudsen phenomenon. We explained regarding the role of RB gene in cell cycle. We mentioned that the tumor suppressor gene is a governor and it's important break in terms of the cancer cell proliferation. We mentioned about the TP53. Remember TP53 we mentioned is a guardian for the genomes and supposed to sensing a damage, trying to fix it, and if they cannot fix it, they do the process of apoptosis. We mentioned if they cannot, and the TP53 gene will become affected, there is risk to get the cancer. TP53 plays a central role in maintaining the integrity of the genome. In non-stressed healthy cell, TP53 has a short half-life time, maybe 20 minutes, because of its association with MDM2. And in this protein, actually try to protect the cells. When the cell is stressed, could be, let's say, as we mentioned, in P53 mutation, and the activated complex induce release of P53 from MDM2 and indicates its half-life. I'll show you how is it. So remember, we mentioned P53 is tumor suppressor genes. Is under which category? We mentioned is guardian, P53.
Yeah.
Okay. Supposed to sensing the gene damage. So P53 prevents neoplastic transformation.
No, it's a governor.
Governor was RD. Guardian was P53. Guardian is a sensing genetic damage. Okay. So p53 mediated cell cycle arrest may be considered the primordial response to DNA. So they try to stop actually the progression of the cell cycle from the G1 to S, because in the S we have a synthesis. p53 mediated cell cycle arrest may be considered the primordial response to damage and re-arresting the cells in the G1 phase. If DNA damage is repaired, we mentioned successfully, p53 actually upregulate transcription of the MDM2 and leading to destruction of p53 and relief of the cell cycle. We said that's become normal again. If the damage cannot be repaired, the cell may enter p53 induced senescence and undergo p53 directed apoptosis. This is a picture that I already showed and explained for you. p53
MDM2 is
MDM2 actually is helping p53 in cell cycle control. p53 induced synthesis in the permanent cell cycle arrest characterized by specific changes in morphology and gene expression that differentiate from the quiescence or reversible cell cycle arrest. And again, p53 also we mentioned if cannot do the process of repair, and that's there is no chance to get the repair, p53 induce apoptosis and programmed cell death, and it is the irreversible DNA damage and cell is going to die. When there is homozygous loss of p53 genes, DNA damage goes unrepaired, mutation becomes fixed in dividing cells, and the cells turn onto a one-way stream leading to the malignant transformation. It means when remember we mentioned for tumor suppressor genes, both alleles supposed to be affected. So we need homozygous loss of p53 to have a malignant transformation. So confirming the importance of p53 in controlling carcinogenesis, more than 70% of the human cancer have a defect in these genes. Actually, nowadays the p53 is very important. When you study the chapter of neoplasia in hematological malignancy, you see that the p53 is very important in terms of behavior of the tumor. When p53 become mutated, we have a variant allele frequency. When the variant allele frequency is too high, more than 50%, this cancer, they are very aggressive in terms of behavior, in terms of progression, in terms of the recurrence and relapse. So they try to classify the tumor, especially hematological malignancy, based on the p53 profile. Is it p53 mutated or not mutated? If it's mutated, it means that the tumor has an aggressive behavior, and in terms of response to treatment is not good, and it's very difficult for the doctor to manage these patients. So I know it is more than 70% of the human cancer have defect in these genes. See, that is very important in all different types of cancer. For example, colon cancer, myeloid malignancy, breast cancer, lung cancer, and all they are related to disease p53. Patients with the rare Lee-Fromen syndrome, remember we had classified as autosomal dominant and autosomal recessive cancer. Syndrome inherit one defective copy in the germline and lost the second one in somatic tissue, like as RB in familial type. Such patients have the chance to get the cancer before age 50, and it means that have more risk of the cancer in the young time, the age. This one we have explained before. Okay. This is the example. Growth inhibitory signal could be different mechanisms. Sometimes this mechanism could be a contact inhibition. The cell, when they contact to each other, they send the signal to stop their proliferation. One of the examples is the cell-cell contract formed in this monolayer. When the cells become monolayer, they send the signal to each other that stop the process of proliferation. This is another mechanism. So for cancer to become insensitive to the growth inhibitory signal, sometimes there is a problem in the contact inhibition. And this is another mechanism for insensitivity to growth inhibitory signal. Okay, So, if I want to summarize my lecture for today, we discussed regarding the process of the carcinogenesis as main role of the cancer. Okay. So the process of the carcinogenesis we classify it as 4 groups of genes. We mentioned we have oncogenes, we have oncogene and proto-oncogene. They are normal. We mentioned they code the protein as oncogene, but when we say oncogene, they are abnormal. They are important in the growth factor receptor, and then we mentioned signal transduction protein, nuclear transduction protein, and all this mechanism we call it the oncogenes. The second group we say that the tumor suppressor genes. Tumor suppressor genes also we say that they are important to control the cell proliferation. The 2 important tumor suppressor genes we mentioned RB and CP53. The RB genes we mentioned is retinoblastoma genes, control the RB protein, and this RB protein actually control the cell proliferation. Another example in this category we mentioned P53. P53 is very important, is under group of tumor suppressor gene and is a guardian. RB is a governor, P53 is a guardian. P53 is supposed to sensing the gene damage and try to repair it. If it can repair, the process of the cell proliferation can continue. If it cannot repair, what we have the apoptosis. If there is mutation in P53, there is no repair, there is no apoptosis, and as a result, we have the cancer and malignancy. The third group we mentioned apoptosis, those genes that they control the programmed cell death or apoptosis. If something wrong happens in these genes, for example, for BCL2, as a result, there is no apoptosis, there is no programmed cell death, and again, we have a risk of cancer and malignancy. And the fourth group, we mentioned those genes that they have contact, for between the tumor cells and host, that they provide the nutrition and blood supply, and this is a process of carcinogenesis. It's very important to know the process of the carcinogenesis in terms of behavior of the tumor and in terms of target therapy. Because if we know, the location of the affected genes, for example, I mentioned chronic myeloid leukemia, because we know there is a fusion between the BCR and ADL1, there is proliferation of the tyrosine kinase. We treat the patient with tyrosine kinase inhibitor. For those people that they have different types of tumor, they have a specific drug, and then we can treat the patient. So it's important to know the base and carcinogenesis of the tumor. The second part of my lecture we discussed regarding the hallmark of the cancer. I just mentioned a few. My colleague Professor Yusuf is going to continue. We mentioned this process of the carcinogenesis and tumor cell proliferation is associated with cell sufficiency in terms of growth signal, and they become insensitive to growth inhibitory signal. They can do the process of the cell proliferation without any signal inhibitor, without response to any inhibitor signal. They escape from the apoptosis, they escape from our immune system, they produce inflammation, they do angio invasion, they can metastasize, they can invade. This is the hallmark of the cancer. I just mentioned a few of these, and then my colleague is going to continue the hallmark of the cancer. If you have any question, I can answer.